Hi, it's Miss Barnhart. Today, let's continue our discussion in genetics by looking at gene linkage. This is another instance where ratios from crosses do not follow expected ratios. But once we understand the behavior of the chromosomes, it all makes sense. In all of the other crosses we've studied, genes are located on different chromosomes. This is the basis of the law of independent assortment. Today, we're going to examine what happens to the inheritance of traits when genes are located on the same chromosome. A dihybrid cross between two heterozygotes has a 9:3:1 ratio. Imagine, though, if the genes were on the same chromosome. How would this affect the outcome? We definitely would not see the independent assortment because in this case, the genes travel as a pair. What happens if the genes are located on sex chromosomes? How does that affect the pattern of inheritance? These two questions will be addressed in today's lesson. The fact of the matter is that some genes do travel together. This concept of linkage was first explored by Thomas Hunt Morgan in the early 1900s while he was studying at Columbia University. He was investigating the genetics inheritance patterns of fruit flies. Fruit flies are a good choice for study because they are small, have a short generation time, and produce many offspring. They also only have 4 pairs of chromosomes. After many breeding experiments, Morgan discovered a mutant male fly with white eyes. When he bred this white-eyed male with a red-eyed female, all of the F1 had red eyes. Subsequent breeding of the F1 led to a 3-to-1 ratio of the F2. However, there was a surprising addition. Only the males in the F2 had white eyes. Therefore, Morgan concluded that this trait must be related to gender. He rationalized that if it were not, then the white-eyed trait would be equally prevalent in males and females. In order to explain this occurrence, let's revisit how gender is determined chromosomally. In humans, as in most sexually reproducing organisms, chromosomes are inherited in pairs. Each species has a characteristic number of chromosomes. In humans, this is 46, or 23 pair. One of each pair of chromosomes is inherited from each parent. Of these, 22 pairs are autosomes, which are homologous pairs. The 23rd pair is different in different genders. Males are XY, while females are XX. Fruit flies have 4 pair of chromosomes, with 3 pair being autosomes and 1 the sex chromosomes X and Y. You can see the female XX chromosomes here and the male XY in these diagrams. Morgan inferred that since the trait only appeared in the F2 males, it was on the X chromosome. X big R is the red allele, X little r the white allele, and Y designates the Y chromosome. Females must receive 2 of the little r alleles in order to express the white-eyed trait, but males will express the allele they receive from the female parent. Let's see how this works with the white-eyed trait. Here are the parent flies, a white-eyed male and a wild female, or red eyes. Can you see that the female parent has 2 alleles for red eyes, one on each of her X chromosomes, and the male has the white allele on his solitary X. Here are the gametes that can be formed. The female will produce only wild red alleles, while the male will produce 2 types, a white-eyed allele on his X chromosome and the Y chromosome. You can see the results of the F1 here. All of the F1 have red eyes. The females inherit a red allele from their female parent and a white allele from the male parent. They are heterozygous, but the red allele masks the white one. Next, when the 2 F1 flies are crossed, all of the females are red-eyed, but half of the males should have red eyes and half should have white eyes. Now let's take a look at what happens if we're to cross a white-eyed female with a red-eyed male. In this case, the F1 males are all white while the females are all heterozygous and have red eyes. Crossing the F1 gives a predicted 1:1 to 1:1 ratio of red-eyed females to white-eyed females to red-eyed males to white-eyed males. The key here is to be sure you're using the XY notation as you complete these crosses. Now it's time for you to try an example. In cats, the gene for tortoiseshell or multicolored coats is codominant and located on the X chromosome. Females that receive a B and an R allele have black and orange splotches on white coats. Males can only be black or orange, but never tortoiseshell. Can you show the cross of a female tortoiseshell cat with a black male? Remember to use XY notation for the trait. Pause the video to work out the problem. Welcome back. Let's check your work. First, be sure you set up a key. It's usually most helpful if you start using the X notation here. Now let's check your parental genotypes. You want a heterozygous female and a black male. The black male only has one allele for color and a Y chromosome. Here is how we set up the Punnett square. And here is how the genotypes are completed. Can you see the probable phenotypes now? 1/2 of the offspring are expected to be black, and these are equally split between males and females. 1/4 are expected to be orange males, and 1/4 are expected to be tortoiseshell females. Any other sex-linked trait that you will set up will be in exactly the same manner. Just use your XY notation. Some common human traits that are X-linked inherited are hemophilia, red-green color blindness, and Duchenne's muscular dystrophy. In each of these, we see more males affected than females, since females must inherit the alleles from both parents, while males need only to inherit the allele from their mother. Additionally, only females can be carriers for the conditions. There are some Y-linked alleles, but these are very few, and in these, all males inherit the affected Y from their father. That's sex-linked inheritance, and I hope that's helpful.